Chapter 8.3 Cloning. Cloning is the process of making an identical copy of an original. In biology, it is used in two contexts. First, cloning a gene involves using recombinant technology. A gene is extracted from one organism and is then inserted into a bacterium where it can be used and studied. In contrast to genetic cloning, which involves making a copy of a gene, biological cloning involves cloning an entire organism. That is, reproductive cloning. Cloning can make it possible for cattle or sheep with desirable characteristics such as high milk production or fine wool growth to be produced more rapidly than through the normal cycles of reproduction and selection. It is achieved by embryo splitting or by nuclear transfer. Embryo splitting. In embryo splitting, egg cells are removed from the donor female and fertilized in vitro, that is, in tissue culture, by sperm from the male. After the fertilized egg has divided, the coat around the two cells that promotes cell division is removed and the two cells are separated. Each cell is then given an artificial coating that promotes cell division. Embryos that have just begun to differentiate, called blastocysts, are implanted into surrogate mothers. The individuals are genetically identical, they are like identical twins but in different surrogate mothers. Nuclear transfer. The process of cloning by nuclear transfer came to prominence in 1996 when Dolly the sheep was cloned. Nuclear transfer involves removing mature donor somatic cells from a mature animal and a recipient egg cell from another mature animal of the same species. The donor cells are cultured in a nutrient medium before becoming inactive, and the nucleus of the recipient egg cell is removed. You can look at this figure 8.7 is going through all of this. The donor cell with the intact nucleus is fused with the hollow egg by an electrical impulse. The new single-celled embryo is cultured for about a week, then cell division is activated and the developing blastocyst is surgically implanted in the surrogate mother. The offspring is genetically identical to the nucleus donor. Cloning using nuclear transfer has not proceeded without some controversy. The success rate of live births is low and many of the offspring suffer from deformities. For these reasons, the scientific world is almost universally opposed to experimenting with reproductive cloning for humans. Application of cloning in agriculture and horticulture. Animal cloning, like other types of cloning, is used to increase the number of breeding animals with naturally occurring desirable traits. This allows for the rapid spread of these characteristics through the herd. The cloning of farm animals for food production is already underway in a number of countries. In the European Union, the EU, a draft directive on cloning of animals aims to ban temporarily the use of the cloning technique on EU farmed animals. The draft directive allows cloning for purposes such as research and conservation of rare breeds and endangered species, or for use in the production of pharmaceuticals and medical devices where the techniques can be justified. Keen gardeners are aware of the ability of plant cuttings to regenerate into mature plants with all the many different types of cells needed for the plant to function independently. The brand new plant is a genetic replica of its parent. It is a clone of the parent. Gardeners may consider regenerating plants by cloning rather than using seeds from the parent plant when the parent plant has a desirable characteristic they want to keep. If the new plant was grown from a seed of the parent plant, the genetic material would not be an exact replica, and the desirable characteristics may not appear in the offspring. As far back as the beginning of agriculture, humans have been cloning plants from cuttings and by other methods. Cultivated olive trees originated about 6,000 years ago in Asia Minor. Somewhere between 3000 and 1800 BC, olive trees were introduced to Egypt from Syria. There are many records of clonal propagation of these trees. Today, many economically important plants are commonly grown using cloning techniques, including vegetable crops such as potato, fruit crops such as bananas, strawberries and dates, and even woody species such as pines and grapes. Cloning can be used to conserve rare or endangered plant species. The Wollemi pine, a plant thought to be extinct and only known previously from fossil records, has been saved through cloning. A small number of the pines were discovered in 1994 in the Wollemi National Park in the Blue Mountains of New South Wales. Ancestors of the Wollemi pine go back to the mid-Cretaceous and possibly even the early Cretaceous period, some 110 million years ago. All the Willamide pine plants currently for sale around the world are clones of the original trees found in the wild. The cloning of plants by plant tissue culture technologies has had a huge impact on the horticultural industry. Using the technique, cells are grown in sterile conditions on a culture medium supplying all nutrients needed. Micropropagation is the method used to produce clones of a plant, often scaled up to mass production. The advantages of plant cell tissue and organ culture technologies include more rapid production of plants, taking weeks instead of months or years. Very little space is required compared to planting in fields, and the plants can be produced all year round. Because cloning produces the same genetics in the offspring, the most desirable characteristics in the plant can be maintained in a predictable way. Maintaining desirable characteristics can increase crop yield and quality while reducing the incidence of crop failure. There are, however, negative consequences of mass growth of cloned or very genetically similar plants or plantations. The Irish potato famine of the 1840s is an example of planting crops that lack genetic diversity. Cultivation of whole fields of genetically identical potatoes left the entire crop vulnerable to pest and disease infestation. During the famine, approximately 1 million people died and 1 million more emigrated from Ireland. Modern agricultural practice often results in a reduction in the number of crop species. Crops are grown over a wide area for a large number of consecutive years. This allows large harvests with minimal labour. When only one species of organism is grown, such as a wheat field, biodiversity is reduced. This type of farming, resulting in a monoculture, a colony of genetically identical individuals, often requires extensive use of fertilizers along with pesticides and herbicides to reduce the natural tendency of the community to diversify. Another downside of this practice is the spread of disease and susceptibility to changing environmental conditions. Cultivated plants grown in genetically homogeneous monocultures do not possess the necessary ecological defense mechanisms to tolerate the impact of pest outbreaks. As a result of reduced biodiversity in monocultures, the natural enemies of pests are no longer present. If pest outbreaks occur, the population of beneficial insects are missing, and the weather conditions favourable to the pest happen simultaneously. The effect on the crop can be catastrophic. Recall. Cloning can be accomplished using either embryo splitting or nuclear transfer and results in the production of an individual genetically identical to the DNA donor. Animal cloning is used to increase the number of breeding animals with naturally occurring desirable traits. Cloning can be used to conserve rare or endangered plant species. Mass production of plants often uses the method of micropropagation.